Hello, and welcome to JDRF's October Research Information Volunteer Call on Cell Therapies. Today's presenters are Esther Latres, Ph.D., Assistant Vice President of Research, and Jaime Giraldo, Ph.D., Associate Director of Research.
Welcome, everyone. The next three RIVs will be focused on cures, and you'll be listening from the JDRF scientists that are leading our research mission of accelerating breakthroughs and therapies to cure type 1. Depending on the stage of the disease, we know that to cure type 1, we need to identify people at high risk of developing diabetes. We need to work on disease-modifying therapies to delay, reverse, or stop progression to the onset of type 1. And we also need to work on replacing the lost beta cells with highly functional cells. So while the priorities may not have changed from last year as we present this RIV on cell therapies today, we'll talk about the progress that has been in the field, as well as how we adapt or modified our strategies and approach to achieve our goals or priorities. So you'll hear from Jaime Giraldo today. So I'm excited to listen to Jaime. Thank you.
Thank you, Esther. Hello, everyone. I'm Jaime Giraldo, and it's great to be here and have the opportunity to tell you about the work that we're doing for cell therapies and the development of cures for type 1. So when we speak about cell therapies, what are we referring to? These are therapies which use live cells to treat or reverse disease. And at JDRF, this means externally derived beta cells, or islets, to restore insulin independence and glucose control. JDRF is accelerating the development of cell therapies towards commercialization through preclinical and clinical testing, and also supporting regulatory approval of these therapies and ensuring that people have access to these therapies once they are approved. Now, we know that cell replacement is effective in delivering cures for T1D. And so we know this through clinical islet transplantation. And you can see here on the figure on the top left, which kind of depicts what clinical islet transplantation entails, which is isolation of islets from a deceased donor, which are then infused into the portal vein or a vein in the liver of the person with type 1, where the cells lodge and cure type 1, result in insulin independence. Now you can see on the bottom a continuous glucose monitor trace of the blood glucose of a subject before and after transplantation, and you can see a dramatic improvement in their glycemic control. And so this is much better than any of the results that people have been able to achieve with all the new technologies in terms of hybrid closed loop systems, insulin pumps, and new insulins. And for this reason, we are pursuing the development of these cell therapies. Now, they are limited to, because of two factors. They rely on deceased donor cells, which are very scarce, and so we don't have enough tissue to treat everyone. And it does require broad immunosuppression, which has unwanted side effects. And so for this reason, this therapy is currently only available for people that are having a very hard time managing their diabetes and have severe hypoglycemia unawareness. Now, in terms of how can we succeed in developing cell replacement therapies, we need unlimited sources of cells. As I mentioned, they are scarce at the moment, and so we need to make sure we have enough to treat everyone. There are a few different sources that we are exploring. Allogeneic sources refers to cells from humans that are transplanted in other humans, so from one human to another, or xenogenic sources, which means from other animals and that are being implanted into humans. And so in our case, the most explored option is porcine islets, given the history of use of porcine insulin in humans, and of course, pigs are widely available. Now, in addition to the source of insulin producing cells, we need a strategy to deliver the cells and ensure that they stay alive and functional without the use of this broad immunosuppression. And so we are looking at alternative sites for implantation of the cells. As I mentioned previously, they are infused into the liver, but the liver is not an ideal site. It's a very toxic environment, which is detrimental to the cell function The cells are in direct contact with blood, which can cause inflammation, and so that could lead to immune rejection. And so we're looking at other sites where we can deliver the cell, for example, the skin, the abdominal cavity and the omentum, or in some cases also exploring skeletal muscle to see if we can deliver the cells there and they can perform their function. We need to make sure that when we do deliver the cells, they have enough oxygen to stay alive. We need to make sure that we protect them from the immune response once they are implanted. And we need to make sure that they can sense the blood changes in the blood glucose and secrete insulin accordingly to really achieve the clinical outcome that we're looking for, which is blood glucose control. Now, we have renewable sources of cells at this point. And so, as I mentioned before, we're looking at stem cell derived insulin producing cells. You can see on the bottom here kind of a schematic which shows the process of differentiation, which refers taking a cell which is basically naive and it's like a blank page, and through culture in the lab, in the dish, trying to replicate natural development in the embryo through different kind of stages of different combinations of factors and chemicals, we can get them to differentiate into insulin producing cells. And as you can see, there are several companies at this point that can make these cells, the insulin producing cells, from stem cells. And so that's been in large part thanks to a lot of funding from JDRF in this space. So we are very hopeful because of the fact that we can make these cells now in the lab at numbers large enough for implantation. Now, some of these companies are looking at testing these cells at different stages. Some are looking at implanting pancreatic progenitor cells, which then mature in vivo, while others are looking to implant cells that are much matured in the dish before implantation. Now that we have the cells available for implantation, we need to figure out how are we going to deliver the cells and protect them from the immune system. And we are exploring multiple approaches to do this. One is encapsulation, which uses a physical barrier to block the immune response while allowing the cells to receive their nutrients and nourishment from the surrounding environment, sense the blood glucose, and then secrete insulin accordingly. We're exploring scaffolds, which are kind of these matrices that are open and allow for housing and distribution of the cells within that space, and allows us to kind of engineer the local environment to keep the cells alive, promote the infiltration of blood vessels to vascularize the site, deliver oxygen if necessary to enhance survival of the cells, and could be also used for delivery of drugs locally to modulate the immune response. And so closely related to that is the approach of immune modulation, where we are trying to use local delivery of drugs or other biotechnologies that can really alter the immune response to these cells at the site. And so this is done in a manner that is localized so that, you know, it's basically an immune privileged site that does not need the whole systemic or broad immunosuppression. And so the person's immune system can work or function as normal, but the cells are protected locally. And then finally, we have gene editing, where we employ gene editing tools to modify the cells and so enhance their survival and function. In terms of the cell therapy roadmap, this is how we envision the development of different basal replacement products. Today, we are at islet transplantation, which, as you can see, is for people who have severe hypoglycemia unawareness. It does use donor islets and requires broad immunosuppression, and this is on the left column here. And so as we move through these different subsequent generations of products, we envision that these are going to require less immunosuppression in terms of the systemic immunosuppression. They're going to be using renewable sources of cells, and then that will allow us to expand the population of patients that can get these therapies, since the requirement for immunosuppression is gradually reduced. And of course, we see that as we go through these different products, the insulin requirements are going to reduce or be eliminated, while at the same time improving glycemic control. And so You can see there, we start off with renewable cell source with broad immunosuppression in this hypoglycemia unaware population, moving to a product that uses different methods, either encapsulation or immune modulation to supplant the broad immunosuppression and a renewable source of cells, and that expands the population to adults. And finally, to a product that really completely eliminates the need for immunosuppression and can be used by anyone with type 1 diabetes. Now, in terms of encapsulation, as I mentioned before, the gap that we're trying to address here is to protect the cells from immune rejection without the use of broad immunosuppression. And so this approach basically uses a physical barrier to block immune attack while allowing exchange of nutrients, glucose, and insulin to achieve a therapeutic effect. And so there are a couple of different approaches for encapsulation. Macroencapsulation, where is a single device, a large device, which can house a large number of cells and is implanted where the cells can function, or micro or nanoencapsulation, where single or a few clusters of cells are encapsulated in these small microcapsules. And so these can be implanted and the cells can perform their function. Now, with the macroencapsulation device, it enables retrieval of the device since it is a single device with all the cells. However, these devices are limited in terms of the diffusion of nutrients and how many cells the devices can house because it's limited by diffusion for the transport of all the nutrients. Whereas with microcapsules, because it's fewer cells within each capsule, there's higher surface area to volume ratio, the exchange profile in terms of nutrients is better, but then we have to implant multiple capsules, and so that makes retrieval more challenging. And so the field has moved also towards nanoencapsulation approaches, which entail coating these cells or cell clusters with thin layers of materials, much thinner than the microcapsules. And so this reduces this barrier for diffusion and helps in terms of the implant volume and the sites where these cells can be implanted. Now, of course, both of these entail using biomaterials to create this physical barrier. And so one challenge is the fibrotic response, which is a natural response to any type of implanted device or material that is foreign into the body. The body recognizes it as foreign and essentially walls it off with this fibrotic capsule. And so that's one of the challenges that the field has faced for quite some time. Now, in terms of research funded by JDRF in this space, we've been funding Bob Langer and Dan Anderson at MIT for some time to address some of these challenges with encapsulation. They have been very successful at addressing this issue by developing some chemical modifications to these biomaterials. And so by creating better biomaterials, we've been able to encapsulate cells, prevent this fibrosis, and really lead to improvements in terms of the glycemic control in animal models. And so because of this work, the scientists founded Sigilon Therapeutics, which is a biotech startup, which is currently pursuing this in collaboration with Eli Lilly to drive this cell therapy work into the clinic and develop these technologies further. And of course, research in novel biomaterials will prove useful for other devices, implantable devices, and we continue to support the team at MIT to further develop other encapsulation devices that are promising. Now, of course, to develop all of these devices, the technology must undergo rigorous preclinical testing before it gets into the clinic and testing in humans. And so we are not only funding the researchers at MIT, but a few other groups across the country and the world, really, to develop these technologies and move them from small animals, mice and rats, into large animals, mainly pigs and monkeys, for testing before going into humans. And so we have different approaches that we are supporting to have multiple shots on goal and increased chances of success. In terms of immune modulation, so just to give a quick overview, the strategy is to really alter the immune response to the implanted cells and or the device, which will allow the cells to thrive without the use of the broad immunosuppression. And so the idea here is to use different biomaterials for controlled delivery of drugs locally and or presentation of helpful immune signals at the implantation site that is going to protect the cells ultimately from that immune response. And so we can also use these systems to re-educate the immune system so that it accepts the cells that are implanted. Now, this work that I'm showing here is actually was originally funded at Georgia Tech and the University of Louisville by Andres Garcia and Haval Sherwan, where they developed these microgel technologies to present certain immune signals locally. And so these microgels are co-transplanted with the islets. It does not involve any encapsulation and resulted in protection and acceptance of the implanted cells. And so they were able to demonstrate prolonged survival of the implanted cells and long-term function when combined with a short-term immunosuppression of only a couple of weeks. And this led to the founding of another startup, a biotech company called iTolerance, which is also pursuing clinical development of this technology. Now, what is really interesting is that these microgels are platform technology, so they not only deliver this fast ligand molecule, which is what they've used previously, but they can also deliver other immune modulating molecules to improve or supplement kind of what they've seen up to this point. In terms of gene editing, the idea here is to employ gene editing tools to enhance the survival and function of the implanted cells. Now, in terms of genes and how does that work, you can think of it as DNA is basically the plans or the instructions for the cells and parts of and the proteins that are basically the machinery that makes the cells work. And so genes encode certain proteins. And so when these genes are expressed, these proteins are made, and then the proteins go and perform whatever it is that they need to do, whatever their function is in the cell. And so by editing the genes, we can edit these proteins and ultimately modulate or affect how these cells function. And so we can intervene by targeting genes or proteins that are involved in the immune system, so immune recognition, activation, autoimmunity, and tolerance. And we can also target genes or proteins that make cells more robust and enhance their function. And so we have been supporting some centers of excellence in this space, and I'm just going to hand it over back to Esther to speak about the centers of excellence.
Yeah, so the ability to engineer cells and use them as curative therapies plays a key role in our centers of excellence. So just to remind you, these centers of excellence bring expertise from different institutions, different laboratories, and put expertise together to just develop the next generation of curative therapies. So the centers basically have many different focus, but one of them is how to protect the cells from the immune system by engineering these cells. So the center from Northern California and New England, for instance, have already developed a way of differentiating cells that Jaime was mentioning. So the next stage is engineering these cells to make them stronger, to make them inner from the immune system as a way to survive and function after implantation. So the centers have some overlapping, which is not, it's good to have some overlapping with the centers. These are still early in discovery, and there are many different approaches and strategies again to address the challenge. But also they have very unique focus. The Northern California is also very focused on how can we engineer some component of the immune system. So they work with the beta cells that are implanted to protect them. The New England is collaborating, or part of the New England Center, a big part of it is how do we develop better laboratory models, both in vitro models and also animal models, to test these cells and understanding how these engineered cells will be implanted and how the immune system will react to these cells. And the University of Michigan is focused on how to make the stem cell derived beta cells have a better function. These cells require a lot of energy to sense glucose, to produce insulin. So they're working on how to make these cells much better functioning under the stress of being implanted. So we believe that the combination of these three centers can definitely accelerate the development of cell therapies.
Thanks, Esther. And so in this space, we are funding several groups. One of them is Dr. John Odrico at the University of Wisconsin, where he is introducing several modifications into these cells so they can evade the immune response. In an earlier grant, they created cells that overexpress these immunoinhibitory molecules and don't express some of the molecules involved in immune cell recognition, and demonstrated that these edited cells can be transformed into insulin-producing cells or islet-like structures that the gene editing did not affect their ability to do so. And so the team is now looking at evaluating how these cells evade the immune response and are able to cure type 1 diabetes in mice. Now, I'll just mention the scaffolds. We are funding research in scaffolds, as I mentioned before. They are these kind of porous matrix, biomaterial-based approaches, which are kind of frameworks which enable engineering of the local environment to keep the cells alive. And so we can use them for a variety of different things. As I mentioned, they can be used to house and distribute the cells. It's important to distribute the cells because if they all cluster together, they'll be competing with each other for nutrients and oxygen. And so ultimately a lot of the cells will die. And so we need to make sure that they're distributed throughout these scaffolds or throughout the site when they are implanted. They have an open structure, so they allow infiltration of blood vessels. And so this is important, of course, because blood vessels deliver nutrients and nourishment to the cells, as well as the glucose that the cells need to sense. And then once the cells secrete the insulin, it needs to be absorbed into the bloodstream so that it can then be delivered to the rest of the body. These scaffolds or biomaterials can be used to deliver oxygen to support the cells and the survival of the cells during the time period when these blood vessels are infiltrating the site. And so it provides this kind of support during that window, as well as can be used to deliver drugs locally to basically suppress the immune system just at the site, at the implantation site, and protect the cells from the immune response. Now, JDRF has a partnership with Cernova, which is a company based in Canada, who is pursuing a scaffold-like approach. And so the idea here of their approach is to basically prime the skin at the site, at the implantation site, before introducing the cells to enhance the cell survival. And so here the purpose is basically to have the infiltration of blood vessels at the site, provide additional blood vessels there before the cells are transplanted, so that once they are transplanted, they will have all the nutrients they need to survive and function. They are undergoing clinical trials right now, and they are showing very promising preliminary results. Of course, this is a phase 1, so it's essentially a safety and tolerability clinical trial. They have not observed any adverse events from this scaffold cell pouch approach. The device is safe and well tolerated. It integrated well at the site and demonstrated the ingrowth of blood vessels. And there are indications of clinical benefit in terms of reduction in daily insulin requirement and C-peptide, which has been observed in these trials. I just want to mention that all of these examples are just a few of those that we are funding in our portfolio, and we do have several other groups and other projects that are working using similar approaches to address the needs to develop these cell therapies. Now, in terms of the JDRF's role, JDRF established the Beta Cell Replacement Consortium some years ago, which has been critical and played a critical role in accelerating all of these technologies and moving them forward. The consortium brings together scientists from a variety of different disciplines, from engineers to stem cell and beta cell biologists, immunologists, transplant surgeons, which is really what is needed to be able to succeed in the development of beta cell replacement therapy. This is a very multidisciplinary approach that we need to pursue to be able to succeed because of the needs from the development perspective. And so the consortium allows for the exchange of ideas, the sharing of data and results, and really to provide a platform for networking of these investigators to establish new collaborations that can really drive progress forward. And it also allows the opportunity of investigators to meet and interact with other people from industry to really kind of translate their technology from an academic exercise into more of a commercial approach. Now, in terms of catalyzing additional investment, of course, JDRF has invested in this space, but we can't do it on our own, and we need to make sure that the industry and the private sector are investing in these technologies to really develop them and take them all the way to the clinic. And we've seen that, that has paid off in terms of our investment, catalyzing additional investment from the private sector. And so we know that Biosite, I think you're all very familiar with, has established partnership with Gore, which invested in their program, and of course have received a lot of funding from CIRM, the California Institute for Regenerative Medicine, Vertex, which acquired Sema Therapeutics, which was the cell source stem cell derived beta cells that JDRF funded some time ago, as well as the T1D fund. As I mentioned before, Sigilon Therapeutics, which was spun out of MIT, which formed a partnership with Eli Lilly. Novo Nordisk has supported a lot of one of our consortium investigators by providing significant funding, as well as establishing a partnership with Procyon Technologies, another startup that was spun out from the consortium, as well as a few others, which you can see here on the slide. Now, additionally, the T1D fund, which is the venture capital arm of JDRF, has also invested significantly in beta cell therapies. And the T1D fund doesn't invest alone, but also with other VC partners. And so we have seen definitely a significant investment in the beta cell therapy space, particularly the beta cell replacement, as a result of JDRF investments and research. Now, in terms of regulatory activities, as we mentioned earlier, it is very important that these therapies, there's a pathway for approval of these therapies so they can actually reach the community and the people that actually are going to use them. So JDRF has been engaging FDA since 2007, just to make sure that they are being updated on the different science and technologies that are being developed for T1D replacement therapies. We've been doing a seminar series on topics which are developed in conjunction with FDA. And so that includes a lot of what we've talked about today: novel biomaterials, scaffolds, oxygenation and nutrient delivery, immune-modulation, just to make sure that they see and are aware of what investigators are working on. And so they have an idea of what's coming soon for approval. Engaging professional consultants as well with beta cell therapies. So thinking about what are the manufacturing requirements for these products, what is the most efficient way of running clinical trials, and so adaptive clinical trial designs for these products. And so really also kind of making sure we are presenting this information to our investigators in the consortium so that they are aware of the requirements to really develop products and commercialize products in the future, hopefully. Now, the portfolio, we do have a pretty good distribution of allocation of funds across different areas of the portfolio. As you can see, cell source is only 3%. And of course, we see this as a good sign because this is a success in terms of, we don't need to fund this research anymore because there are companies out there making these cells at an industrial scale and that type of quality. And so we are focusing on the methods to deliver and protect the cells from the immune response. You can see there's encapsulation at 25%, graft survival, meaning vascularization and oxygen so that the cells survive at 24%, And then alternative methods of immune protection is quite a big chunk of the portfolio at this point, since these are new technologies that are very promising. And so we want to make sure we explore those. Encapsulation has been supported for quite some time. And so we are prioritizing later preclinical testing and clinical testing of encapsulation. And we are supporting to a larger degree alternative methods of immune protection, which include earlier discovery efforts and preclinical testing. And then also we have our bucket of accelerating clinical translation, which provides kind of infrastructure and other resources that can help accelerate the development of these therapies and get them into the clinic. In terms of clinical trials, there are a few clinical trials that are recruiting currently. I think most of you are familiar with Biosite, which are testing their VCO1 PEC and CAP or VCO2 PEC direct products. They use pancreatic progenitor cells in their devices. Excuse me, the Vertex trial, which is testing stem cell derived insulin producing cells, which are being infused into the liver and require immunosuppression. Again, the PEC and CAP is an encapsulation product that does not require immunosuppression, and PEC direct is an open device that does require immunosuppression. The Cernova cell pouch, as we spoke about earlier, the scaffold, which induces vascularization of the site. The UCSF program, which uses parathyroid gland co-transplantation with islets because these parathyroid gland induce vascularization, so enhance survival of the cells. And a few different trials at the University of Alberta testing immunotherapies, either through polyclonal Tregs or anti-CD40 ligand, which is just an immunotherapy, as well as a new trial that was just posted on the site using a deviceless site. So this is another approach where they try to pre-vascularize the site before implanting the islets to promote their survival and function. All of these trials at the moment are limited to people with severe hypoglycemia unawareness. Again, they do require immunosuppression with the exception of the PEC and CAP trial. And if you would like to see additional information about what trials are out there, you should go to the JDRF Clinical Trial Connection website, which you can see here, or you can just Google JDRF Clinical Trial Connection to get there and look up what are the different trials that are ongoing. So to summarize, clinical islet transplantation has demonstrated that beta cell replacement can provide a functional cure for type 1 diabetes, but it is limited by scarcity of donor tissue and the reliance on this broad immunosuppression. And so we need to address those two before this can become the standard of care. We now have a renewable source of cells, insulin producing cells made from stem cells, which are being tested in clinical trials, mainly the Vertex trial, as well as Biosite. Now our current focus is on methods to deliver the cells to ensure that they remain viable and functional, as well as on methods of how to protect them from immune rejection. And we are exploring alternative sites for transplantation aside from the liver, and methods to deliver oxygen and enhance the vascularization of the cells so that they survive and function, and multiple avenues to protect the cells from immune rejection, mainly encapsulation, immune modulation, and genetic modification of the cells. And so we continue to catalyze progress and accelerate progress through the consortium, which gathers the world-leading scientists in the space, as well as industry partners to accelerate progress. Our investment has led to significant additional investment from the private sector to develop these technologies, and we continue to work with regulators on a path for regulatory approval of these therapies. And finally, we have several clinical trials that are ongoing, which is really promising. We're very excited to see the results because we are going to learn a lot, which we can then implement in subsequent research. And so with that, I will close.